Okay, hopefully today my system will support the videos, so maybe we can watch those videos in class itself. Right? Okay, I will just run through these since we've done it multiple times. Right, so in the history part or in the first part of the chapter, which is basically the first paragraph, you need to remember these 4 points mainly. First thing, these questions usually come in the form of MCQs or one marker questions because it's just name the scientist, right? So Robert Hooke was the first scientist who observed cork under a microscope, and he noticed that there were these tiny cells. So cells are basically structures where these monks used to stay in, like a small. There would be like a small bed and a window and uh, maybe something to keep store food, etc. Right? So that used to be called a cell. So he thought that this structure resembled that cell, and he gave it also the name cell. That's how the name cell came into picture. It is Latin for little room. Okay. Yeah. So yeah, this question is asked a lot of times as an MCQ. Who was the first person to observe cells or who coined the term cell? So it would be Robert Hooke, right? And then there is one more scientist called Anton von Leeuwenhoek or simply Leeuwenhoek, right? Be careful of this spelling. It's double E and Hoek. So Leeuwenhoek discovered free living cells with the help of improved microscopes. Leeuwenhoek was basically involved in making microscopes. So he would experiment a lot with different types of microscopes to see things better, right? So he was able to see the free living cells properly. Here, the reason they have written free living cells is cork cells are basically dead. So Robert Hooke, though he was the first person to see the cell, he observed dead cells, whereas Leeuwenhoek was the first person to see the living cells.
Ma'am, and it was like pond water, so free living cells.
Correct. So Leeuwenhoek actually he had this habit of seeing everything because he was involved in lenses and microscopes, right? So he would take anything or used to roam around with his microscope and observe every single thing, how it looks or what is it, like that. Yes, so that's how he came across the free living cells. Then these two terms, these two scientists, for them, you don't need to remember how they actually discovered nucleus or how did they come across these terms. But yeah, you couldn't just remember that Robert Brown discovered nucleus and Perkins J coined the term protoplasm. Okay. The next important part would be the cell theory. This is again, yeah, like you must have seen, you were already asked in your paper. So this is a question which could come, which is the statements of cell theory, right? That is most commonly asked. So two scientists came up with the initial statements. The scientists were Schleiden and Schwann, okay, and later expanded by Virchow. So these are the three statements of cell theory, where the first two were given by Schleiden and Schwann, and Virchow added that all cells arise from pre-existing cells, right? Okay. I think there is, in case any of you would have missed our first class on cells, we had watched a video where a TED-Ed video on this topic, and yeah, this is the video here. So maybe after class you could uh, watch this video. It's interesting, right? They basically talk about the history of cells in this. It's a small video, just 3 to 4 minute video. Okay, second important part in the from the introduction part itself is the compound microscope. Again, this is usually asked in practical based questions or sometimes as a one marker or just for labeling. It's not asked in detail. You're not asked the principle or how does it work or anything. You just need to know the basic diagram and labeling, right? So yeah, let's just go through it one by one. So the eyepiece is the topmost piece here. Basically, this is where you know your eye will be here. So you'll be bending down like this. So this is your eye. Let's say, sorry for the scribbling. So you'll be looking into the specimen from here, right? As the name suggests, that's why it's called the eyepiece. Okay, and this is where you basically keep your slide or anything you want to observe is placed on the stage. So this is how your slide looks like. It's a rectangular slab and there will be a dot or a very small amount of whatever you want to observe will be placed in the center and covered with a small glass square, right? It's called a cover or yeah. Okay, Sujal is saying eyepiece is also known as ocular lens. Yeah, correct. So ocular basically is anything which refers to eye, right? Correct. So yeah, you can also remember it that way. Okay, so this your specimen will be mounted on the stage, right? You hold it in place with the help of these clips. There will be multiple clips to hold it in place. Okay. Then you can come to adjustment knobs. Right, so it's basically like something like maybe I don't know if you've done this anytime. Usually when we have or when we want to listen to the radio, we need to tune it to a particular frequency, right, to hear it properly. I'm not sure. Has any one of you done that? Have you tuned a radio or once? Okay, Sujal says once. Okay, let me think of some other example. Uh, what else? Okay, have you ever been to an optician for an eye test? Have you got an eye test done anytime? Maybe that would be relatable.
Yes, ma'am.
Okay, so looks like that is relatable. Okay, Sujal also said yes. Okay. So when you go for an eye test, basically there is this chart of letters, right? In front, there will be big letters and then they keep decreasing as you keep going down that screen, right? Yeah, and yeah, it is inverted. Correct. Yes. So basically, the point I wanted to tell you here is that when you are reading that chart, the person or the optician basically keeps changing the lens, um, right? He keeps asking you whether can you see better with this or can you see better with this lens, right? That process is repeated till you are saying that, yeah, this is clear, right? So this that similar kind of process happens in the microscope itself. Any microscope you take, it can be a simple microscope or a compound microscope. A compound microscope is used commonly used one, right? So we need to adjust everything so that you can look at the specimen properly, right? That's all these things help you to do. So once you've understood that part, remembering what they exactly do would be easier for you. So think of it like that. If you haven't really been to an eye test, what you could do is after class, I'm sure all of you have access to a phone, right? You could try switching on the radio on your phone itself and then check. So I'm not sure. I think there is an FM station called 93.5. So if you go beyond before this, like go to 92 and you'll be able to hear only static, right? You don't have it on phone. Okay. I think you might just check in your tools section of your phone. You might definitely be having one. I don't think they have discontinued it yet. Okay. Yeah. So there might be an option where you could, move this, go down, or you could. Okay, you're talking about the frequency one. I think you still have it. Just check once. I'm not sure my phone has it and it's not that old. I mean, it's quite a recent one. So just check. Okay, you don't have to adjust frequency. ठीक है, then let me just skip this part. ठीक है, cool. I was just giving this as an example or to help you understand what how a microscope functions, because right now you might not have practical access to it, right, to view a microscope, to hold it, or etc. Once the lab starts, or you are back to school, you might be able to have a look at it. ठीक है. We had it in 7th. Okay, then ठीक है. If you've seen it before, okay. Yeah, so basically all these screws, so coarse adjustment, fine adjustment, the objectives, all these three um, parts help in basically giving you a better view of your specimen. So first, there are three lenses here. So it's one is 10x, one is 50x, and one is 100x. This is the for a normal compound microscope. That's how it is. Based on the, there are advanced ones, or based on what you are trying to look at, there might be different lens available. Like it might go beyond 100x also. Okay, so first you observe it with 10x. Once you've adjusted everything with 10x, then you move on to 50x. You adjust it even more, and only for specific things like you're observing very small cells or very tiny organisms, only then you go to 100x, or else it's always just 10 and 50, right? That's what objectives are. They are basically, they help to focus and enlarge the specimen, right? And coarse adjustment screw basically helps you move this whole thing up and down. Okay, these two screws help you move this whole thing would be the eyepiece and the objectives. They help you bring it down or bring it away. Is amoeba seen in 10x? No. For amoeba you need 100x. Yes, amoeba is very very small, right? Yeah, you would need 100x. Okay. Yeah. Don't worry. Maybe in 10th, or maybe after that, maybe in 11th or something, you might get a chance to observe blood cells or commonly seen cells, maybe cheek cells or onion root tips or things like that. You will be able to observe. You will even get to observe mitosis or cell division happening, right? It's a real beautiful thing to observe under the microscope. Okay. Yeah. So the coarse adjustment helps you move this whole thing up and down, and the fine adjustment screw also does the same thing. It's basically at a finer level. So coarse adjustment would go up and down really fast, or too much larger distances, whereas fine is when you just want to adjust it to some millimeters, right? You could think of it like this. Okay. So that's for fine adjustment. So they perform the same function, right? Okay. A mirror is used to reflect light into the objective lens. Yes, correct. So the light would hit like this ideally, and you're basically hitting the specimen with light from below. Okay, this part here is transparent, or it is also a glass pane. So the light hits it from below, and that's how you're able to see clearly. Yeah. Okay. Yeah, so that's mirror, and there's a base to support. So this is the base. It's not marked in the figure. This is the base. It's used to support the whole thing. Clips to hold the slide, like I mentioned. These are the clips. Okay. Condenser to focus the light on the specimen. Okay, yeah. Uh, there would be a condenser somewhere near the mirror itself, right? Okay. It's again not mentioned here, not given here also. Lens tube to hold the lenses, these would be the lens tube. So the lens is inside this black thing. So that would be the lens tube. And arm for carrying the microscope, this is basically your arm. Okay, so this is the basic. You could also watch this video where all these parts are again explained in detail, right? Okay. Yes, so there are different types of organisms, right, based on the number of cells they have. So unicellular basically refers to one cell. Uni is one, multi is more than one, right? So these are few examples of unicellular, these are unicellular examples, right? Paramecia, amoeba, bacteria, yeast, and these are examples of multicellular. Yeah, like trees, jellyfish, humans, insects, dolphins, fishes, etc. Right? Okay. Okay, yes. So first thing you need to know is that every cell has okay, Sujal has a question. Two cells will be considered multicellular. Yes. Yes. So there are only two terms, either unicellular or multicellular. Right? So if it is not one cell, it will definitely be multicellular. Okay. ठीक है. Let's go through this a little faster so that we can focus more on the organelles part. Okay. Maybe if you have doubts or anything, you can stop me. Otherwise, we'll just skim through these things. Okay. So basically, every cell, irrespective of the fact it is unicellular or multicellular, has three features, right? Or you could say three parts is not the correct term. I mean, features only would be correct here, or characteristics maybe, right, which are basically as given in the figure here: plasma membrane or the cell membrane, the nucleus, and the cytoplasm. Correct? Okay. So the nucleus or the nucleus and cytoplasm combined give you the protoplasm, right? Okay. So first let's talk about plasma membrane. Yes, Sujal, awesome. Yes, plasma membrane is semi-permeable and its basic job is to regulate the flow of substances. Correct. So this is in short all you need to remember. Again, I think I had mentioned this while we were doing tissues also, right? Similarly, for this chapter also, remember things like that. For every organelle or part or feature that you are studying, always remember the location and function. Right? So if you are a person who likes making notes, or if you prefer studying by making notes, make sure you have made notes like this. So for everything, so you're writing plasma membrane, first write location, then write function, right? Then next you write cell wall, location, function, right? If you're going to do this for the whole chapter, it will really help you. And during exams, then it would just become a one-page read, right? You don't have to search for points or anything like that, right? Uh, the same format has been mentioned in the slides also. Like I've also tried to keep it that way, but I haven't written location and function, the word specifically somewhere, right? So you just have to understand that. So this is the location, right, from here, and function is in the next page because we also have some extra details here, right? So not every organelle has characteristics or composition mentioned. Only where it is mentioned you need to remember, right? Okay. Yes, so plasma membrane is known as plasma lemma or cell membrane. These are the other two names. It's the outermost covering. This is the location. Characteristics: it is living, elastic or flexible, and ultra thin, right? And it's composed of lipids and proteins. Yeah, like you saw, you got a question, right? What it's composed of. So they do ask questions like that. So yeah. Okay, and this definition is important, which is basically the definition of what is a selectively permeable membrane. So you will come across three terms in this chapter. One is just permeable, right? This would be one. Second one is when you read osmosis, you come across something called semi-permeable, right? And the third one is this, which is known as selectively permeable. They're all different terms, right? Where selectively permeable basically means it permits the entry and exit of only some kind of materials and prevents other materials to do so, right? Okay. Semi-permeability, okay, Sujal is saying semi-permeability of cell membranes enables the cell to maintain a constant internal environment. Yes, Sujal, yes. So that is like an implied function, correct? So their main job is to regulate entry and exit because mainly because they don't want unwanted things or they don't want attack on the cells, right? That's why they are They are just like guards, right? They don't, or maybe you could say soldiers, right? You could, the analogy for plasma membrane would be basically guards or soldiers at the front or at the borders, right? They don't really allow anybody and everybody to enter, right? Okay, that's the same thing. Okay. Yeah, so here are the functions, right? This is, yeah. Or you could remember using this cartoon. Yes. Secretions and waste leave. Absolutely, Sujal. Yes. Yes. That's what exit, right? So this is basically an example of what the cell membrane does, right? It allows, this is just one kind of example. Don't keep it stick to that no salt is allowed. Some salts are allowed, some salts are not allowed. That in detail you might learn later if you take up biology, right? Okay. So main job is to provide protection, right? You don't want unnecessary substances to enter or exit, right? And transport, correct. So nutrients will go in and waste products go out, and it acts like a mechanical barrier, right? Okay. Again, this is also a small video which gives you a recap of plasma membrane, and they have also correlated it to a toll plaza or a toll gate, right? So when you're changing states or when you're traveling across the highway or by vehicle, you will encounter toll gates, right, or toll plazas. So that's what it's been compared to. Again, you could watch this after class or anytime you would. Yeah, okay. Right. So then it brings us to this next topic, which is cell transport, or how do substances enter and exit the cell, right? So mainly there are two methods, diffusion and osmosis, right? And these are the examples, right? So most plants gain water through osmosis, and diffusion helps in exchange of gases and water. A commonly asked question usually is differentiate between diffusion and osmosis, right? That is the most frequently seen question. So when you are asked that, you could give the definition as your first point. You could give the presence and absence of semi-permeable membrane as your second point, and an example as your third point, right? So the example is mentioned here, right? Okay. Coming to the definition, so this is basically diffusion. Again, this you would have also done in chemistry, right? Few parts overlap uh, between chemistry and bio, right? Yeah. So diffusion, yes, correct. Yeah. The definition is basically it's the net movement of anything. Anything here refers to solid, liquid, or gases, right? It occurs through all three phases of matter. So anything from a region of higher concentration to a region of lower concentration. It is faster in gaseous phase than in liquid or solid. And here are some examples: movement of oxygen, carbon dioxide, etc. So this is how this is a visual representation of diffusion. Right. I'm not going into detail because you would have done it in chemistry also, right? It's the same thing. But only thing here we'll be talking more about gases, right? Because mainly oxygen and carbon dioxide exchange happens in plants mostly, or yeah, you're just studying diffusion in plants. Next year you would study diffusion as breathing in humans, right? Okay. And we have osmosis, right? Osmosis is a special case of diffusion. Yes, why? Because it's the same thing, and according to chapter notes, it says diffusion occurs in both gases and liquid, and no solid is given. Yeah, because it's rare to see in solid. It's not that it doesn't happen, it's rare to see. So, yeah, at this stage, even if you remember it happens in both liquid and gases, is also fine. Okay. Yes, so osmosis is a special case of diffusion where you have water molecules, only water molecules move from higher to lower concentration, and there is a selectively permeable membrane or a semi-permeable membrane involved, right? Okay. Vacuum cleaner can be a process of diffusion. Not really, not really. No, no, no. I don't think that can be considered an example of diffusion because
Ma'am, because the solid particles move from a region of higher concentration to a lower concentration. Because of vacuum, there is low concentration inside the tube. So,
no, 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 no. No, you actually can't correlate it to diffusion. Why? Because the tube is only creating vacuum, right? It's not creating a concentration difference at all. Inside the tube also, so multiple things get sucked inside the tube at once, right? So you can't really say that it's a concentration gradient like that. It's only because vacuum is created, whatever is there gets sucked into it, irrespective of whether it's solid, liquid, or whatever it is. Okay. Yeah. So, movement of water molecules through selectively permeable membrane till equilibrium is reached, right? So these are the two types of osmosis which is commonly seen. Again, endo always refers to inside. You break down the words, right? Always, which makes it easier for you to remember. Endo is a word which means, or the root word for it would be inside, right? Whereas exo usually refers to outside. So endosmosis is water molecules enter inside the cell. Exosmosis is when water molecules leave the cell. Or you could remember exosmosis is like exit, right? Okay. They basically would leave. Here you have the definitions also mentioned for both in the diagram and in the examples also, right? Okay. Yeah, this is the part where you might get confused or which requires the major amount of uh, what should I say? Major amount of understanding part when it comes to the chapter, right? The all other things are theoretical. There's not much of things you need to understand, but this is the only part where you are asked application-based questions, and you need to get this clear, right? So tonicity, basically the definition is the relative concentration of solutes dissolved in a solution, right? Again, you are doing chemistry, so you must be familiar. There are a solution has two components, right? Basically, it would be we could use the trick. Yes, yes, Sujal. We will come to our trick in a bit, right? So a solution. This is a solution which is made up of two substances, right? One is called the solute, or which is the solid particle usually, right? Usually, and the liquid medium is usually known as the solvent. So these two together make up a solution, right? Again, something you might have done in chemistry already, or you might do in is matter around us pure, right? Okay. So once you're clear with that, these definitions can be broken down and understood easier in an easy manner. Okay. So based on the tonicity or based on the relative concentration, what does relative mean here is always there will be two things to be compared. This you cannot say a solution is hypotonic always or hypertonic always, right? It is always in comparison to something else. Something like saying maybe something like saying you have an apple and a mango, or apple and an orange. Let's take an example of an apple and an orange, right? Only when you eat both can you say that apple does not really have a taste, right? But if you are going to eat apple alone, you will definitely not feel that way. You'll feel that it does have a taste of its own, right? Versus if you take apple and something more bland than apple, or if you're just taking maybe let's say saltless food, if you're eating it with apple, you'll find that apple has more taste, something like that, right? Yeah, yeah, Sujal, you could use any fruit as comparison. I just took an example of something bland and something tangy, right? Yeah, you could use speech also. Think of it like that. So that's what relative means, that there is always two things which are being compared. Okay, yeah, and based on that you have these three definitions. One is hypotonic, where hypo is always stands for low. As you go ahead, you might come across other terms which means hypoglycemia or hypothermia or things like that, right? So always hypo will stand for low, low solute in this case. So if a solution has low solute, here I'm saying a solution has low solute, which means is it going to have more solvent or less solvent? So this is a normal solution, okay? This is a normal solution, for example, and this is my hypotonic solute, where I'm saying it has low solute amount. So will it
High
have,
solvent.
okay, high solvent? What about the others? I have got chat answer. Yes. Sujal, yes, we are coming to that. Coming to that in a bit. Yes, yes, Suhani. So basically, low solute would be it would have more solvent, right? So this refers to dilute, which means it has more solvent, right? That's why we call it dilute. Okay, more solvent or more water. Since we are going to be using these terms only for osmosis, you could directly say more water also, H2O. Okay, right. Or lower concentration of solutes than another solution. So this is the definition. If you broken this down, you could remember it like this. Or other, you could just call this other instead of another. Right? So when you are comparing two solutions, if one solution has lower concentration of solutes or it is more dilute, it's known as a hypotonic solution. Similarly, you can break down the other two words and the same thing has been written. Right? The definition also has been mentioned here. ISO basically stands for same, hyper stands for high or more, right? Okay, and the definitions are given. Okay, moving on. Yeah, now we come to what happens when you are subjecting anything in a hypotonic solution, right? Yes, here is where we were talking about the trick, right? Okay. So when you're placing something in a hypotonic solution, so once you're clear with what hypotonic, isotonic and hypertonic are, now you'll be putting substances in that solution. Like for example, you're placing a cell, right? When you're placing a cell in a hypotonic solution, the medium surrounding the cell has higher water concentration than the cell, right? Since we already said this is dilute, right? Let me write it here, which means it has higher amount of water than the cell. So in osmosis, as you know, water moves from higher to lower concentration, right? So if there is more water outside than inside, the water will go inside the cell, right? Okay. So water moves into the cell. And what is the effect on the cell? The cell swells, correct? Similarly, isotonic means both have the same concentration, same water concentration, right? Inside as well as outside, right? So what happens here is the water moves in and out of the cell at equal rates. Because it moves at equal rates, you will feel as if nothing has changed, right? No change. It looks like no change, correct? Cell remains the same. Whereas if you place it in hypertonic solution, which means here the solute concentration is high, which means it is having less water, right? This has less water. Less water means the cell has more water. So the water will exit the cell and move into the solution, right? Or the cell basically would shrink. Okay. Again, this is a detailed way of just explaining it to you. Once you are familiar, like once you've read this concept multiple times, you would be able to just, you could just ignore all this completely and you would remember what happens when a cell is placed where, right? Okay. Yeah, at this point, I just wanted to mention that read the difference between diffusion and osmosis. I just mentioned in the previous slide a few differences. You could also go back to your chapter notes or the textbook and refer to the differences, right? Any difference or any 3 points are good enough. And this is a cartoon for you to remember what happens when. And yeah, we forgot our trick. So this is our trick also to remember, right? When the cell is in hypo solution, will it swell or shrink? Tell me quickly. Will it swell or shrink? Yes, Sujal has answered. What about the others? Are you all there? Okay. What happened? You forgot what? You forgot the trick or you?
Ma'am, can you repeat the question? I was having some connectivity issues, so I couldn't listen.
Sure, not an issue. I was asking that we have a trick to remember what happens to the cell when it is placed in a hypotonic solution or in a hypertonic solution. So, could you tell me what happens to the cell when it is placed in a hypotonic solution? Does it swell or does it shrink?
Well,
Swell. Yes, correct. So, cell will swell, right? So, our trick is like this: there is an O in hypo, right? Which means you need to remember. Okay, now I got answers. Okay. Yes, yes, Ashka. Yes, yes, Samruddhi. Yes, yes, Sujal. Correct. So there is an O in hypo, which basically refers to a cell which is being swollen, right? It's a complete cell. A cell looks like this. So O would refer to it getting swollen or swelled up. And whereas in hyper there is an E, which means the cell was initially like this and now it has shrunk to this, right? So that's why the here you will say the cell will shrink. That's our trick to remember what happens to the cell, right? Yes, correct. Good. I'm glad you all are answering. Okay. So this is our easy trick in case any of you have missed out or you've not written it or anything for any reason you've missed out on this, you could take it down. Hypo always, if a cell is in hypotonic solution, it will always swell. If a cell or anything is placed in a hypertonic solution, it will always shrink, right? No exception. Okay, right. Okay, we are skipping this and going ahead. This is just, okay, you could go through it after class and check out your understanding. Right, so like I mentioned, again, I have highlighted the location functions here, right? So cell wall is found only in plant cells, right? And where is it found? It's found outside the plasma membrane. It's composed of cellulose. It's freely permeable, and these are the functions. Basically, it provides structure, strength, and protection. Mainly, it provides structure and strength, right? Oh, I've missed out on the R here. It's strength, STREN. Right, protection not much because it allows the entry of everything inside. It's not regulating anything. It's freely permeable, right? So it allows everything to enter. Then at the next stage, at when the substance encounters the cell membrane is when all the checking happens and things are, you know, either sent inside the cell or sent back. Right. And yeah, this is the important definition you need to remember when you are studying cell wall is plasmolysis. Like you have application based questions on osmosis, the ones you saw in your test, right? Similarly, you get a lot of question on plasmolysis also. They are both the same concept. There is no difference. The only thing different would be that plasmolysis occurs in plant cells or cells which have cell wall, right? And osmosis occurs in animal cells or generally in general. Plasmolysis only is pertaining to cells which have cells wall, which would definitely be plant cells, right? Okay. So the trick question they usually ask is they might give you an animal cell, for example, and they might ask you that does this cell ever get plasmolysed? So the answer is no, it can't. It doesn't have a cell wall, so plasmolysis will never occur there. Right. And what does this term mean is basically when a living plant cell loses water through osmosis, right? So the entire cell contents shrink or contract, right, away from the cell wall. That's called plasmolysis, and the cell is known to be plasmolysed, right? Here, this is a basic slide for you to understand what is Turgor pressure and wall pressure. These two pressures are what keep the cell in place and prevent it from shrinking, right? So the cell wall exerts pressure on the contents of the cell. Contents of the cell is basically the cytoplasm and all the organelles floating in the cytoplasm, right? So the cell wall exerts some pressure on the contents of the cell, and in turn the cell contents exert pressure on the cell wall, right? So they are balanced. Because they are balanced, the cell is in shape and it is in place, right? It does not really burst, or it does not move away from its place because these forces are balanced. However, when there is any situation where the two pressures are not balanced, or where the pressure from the wall pressure exceeds the Turgor pressure, you would notice the cell contents shrinking away, or basically plasmolysis happening. For example, this is your plant cell, right? Okay. And because there is too much loss of water outside, there is water loss, right? So the cell or the contents of the cell have shrunk, right? Or they are not able to exert that much pressure on the cell wall as before. Let me just draw cell membrane here, right? So this is your cell membrane, right? And now it is moving, water has moved outside. So what the cell becomes would be something like this. And let me draw the cell contents, right? So basically all the cell contents, they shrink and they move towards one part, any one part of the cell. It's not fixed which part they should move to. They just move to one part because they are not able to hold themselves in place, or hold or spread evenly over the entire surface, right? So this is what is called a plasmolysed cell, right? Okay, I'm not writing the complete thing. Yeah. Okay, so this diagram basically is for you to understand why plasmolysis occurs, or what is the meaning, or how does it happen, or basic mechanism of it. Right? Again, you are never asked to draw this diagram or to explain wall pressure and Turgor pressure. This is for your understanding so that you understand plasmolysis better. Okay. Right. So this is here is a diagram for plasmolysis. Again, like I mentioned, it's similar to osmosis. The only difference is this happens in plant cells. Right. So this is how a normal cell looks like in the first picture, and when you put it in a hypotonic solution, it swells up. Right. So that's how it looks. Right. It's become a bit bulgy. And when you are placing the normal cell in a hypertonic solution, all the water moves out, it shrinks, and the cell contents move away from the cell wall, or it becomes plasmolysed. Right? The third figure is a example of a plasmolysed cell. Okay. Okay, now we come to nucleus, which is our third and final feature of every cell, right? It's also known as the brain of the cell because it controls all the cellular activities. It's also known as the center for storing heredity information, right? Because the nucleus will dictate what happens to the daughter cells when the cell divides, right? It holds all those information, what to pass on, what has to be transmitted, all that is stored here, right? Okay, and the structure is basically it has a double layered covering which is known as the nuclear membrane, right, or nuclear envelope in the figure as shown, right, and there are some pores on it. The membrane is similar to the cell membrane or plasma membrane, similar in structure and similar in function, right. These pores help materials move in and out of the nucleus, right. Okay, and what does it contain? It contains chromosomes. Here, one important difference you need to know is what are chromosomes versus chromatin. Again, this is usually asked towards the end or when you've had multiple tests on this chapter, and this is like a higher order level question which is generally asked. Is difference between chromosomes and chromatin, right? Chromosomes basically contain information for inheritance of characters in the form of DNA or genetic material, basically, and they are made up of DNA and protein. DNA basically contains information for constructing and organizing cells. And here you have a definition for genes also. What are genes? Basically, they are functional segments of DNA, right? These 3 points are where you have the difference between the chromosome and chromatin. If you want me to show through a diagram, this is what a chromosome looks like, okay? And this is what a chromatin looks like. So this thread-like structure or thread-like loops eventually condenses to form this. That's all is the difference. Okay, they are one and the same thing, but usually when a cell is not dividing, the DNA is present as chromatin material or as thread-like structures like this. Okay, and whenever the cell is about to divide, the chromatin material gets organized into chromosomes or structures like this, X-like structures, right? That's the only main difference between the two. They are one and the same thing. They are made up of the same thing. They store the same things. But only thing is, when the cell is not dividing, it DNA is in the form of chromatin. Whereas when the cell is dividing or about to divide, it's in the form of chromosomes. Okay. Okay. Yeah, these are some of the functions of nucleus. Right, you could use this cartoon to remember also. Genetic information bearer of the cell, right? Yeah, main roles are cellular reproduction and determining the way the cell will develop. What I have written in bold, those are the keywords, right? Okay. Going ahead, there is also something known as a nucleoid, right? Again, this is also asked as a standalone question where they might just ask you what is a nucleoid or where is it found, right? So nuclear region of the cell may be poorly defined. Poorly defined refers to the fact that it does not have nucleolus, or it does not have chromatin and chromosomes packed in the way it is in the nucleus, right? So this is chromosome and chromatin, right? That's what poorly defined means, that the components or the parts of the nucleus are not a given detailed structure and function. It's just a mass of things doing all the functions together. Okay, and yeah, main difference to note here is that nucleoid does not have a nuclear membrane, right? And examples would be bacteria or usually prokaryotes, right? Such organisms who lack nuclear membrane are known as prokaryotes. So cells are divided into two categories, eukaryotes and prokaryotes, based on whether they have a well-defined nuclear region or they do not. So the cells which do not have a well-defined nuclear region or who have nucleoid are called prokaryotes, versus the ones which have well-defined nucleus are called eukaryotes, right? Okay, yeah. Uh, so for prokaryotes, there are limited examples which you need to remember, like bacteria, etc. For eukaryotes, there are many examples like humans, animals, etc. are all eukaryotes, right? We all have a clear-cut nuclear membrane and well-defined nucleus, right? Okay. These are some of the other functions or other characteristics of prokaryotes which are given here, right? And this is an example of how it looks like. So the nuclear region is spread out throughout the cell. It is not enclosed within a structure like this, right? Okay, that's all. Yeah, and at this point you could go back to your chapter notes or textbooks and read the differences between prokaryotes and eukaryotes to get a better idea, right? Main thing, the differences would be that one will have and one will not have, and the not have one is always the prokaryotes. Eukaryotes have, right? Char usually is like a membrane, a word for a membrane, right? Pro is like does not have, and eu has. So prokaryotes will always lack everything the eukaryotes have. That's an easier way to remember, right? And cytoplasm is the fluid content inside the plasma membrane. It contains many cell organelles, right? As you can see here. Excuse me. The significance of membranes can be illustrated with the example of viruses. Okay, so this is a sentence which is given as it is in your NCERT book. I have written it here to explain it to you so that when you're reading the textbook you should not feel lost that this sentence does not make any sense or I'm not understanding what it means. For that I have written it here to explain it to you, right? Okay, so basically what it says here is that you can use viruses as an example to understand the importance of membranes, right? Why membranes are really important. A classic example is you could take an example of COVID right now, okay, to understand the importance of membranes. So I'm not sure if you are aware or if you are up to date with the news, right? If you are, you would have heard that they have come up with different variants of COVID-19, which are basically these four as of now: alpha, beta, gamma, and delta, right? Delta is the one which they have found in India and which is not going away easily, right? The other three are in control. So why are there so many variants of coronavirus? Because of this, right? Viruses don't have any membranes and hence they do not show any characteristics of life until they enter a living body and use its cell machinery to multiply. So virus change their genetic material or their uh, information based on what organism they have attacked, right? And so we all are unique. Like we have, even though 99% of our DNA may be the same in species, like what I'm saying is that 99% of DNA between you and me is the same, but 1% is different, and that 1% is enough for us to be called unique individuals, right? So virus, they attack different organisms. They are able to attack like a variety of organisms. They don't really have any restrictions. Why is because that is a huge thing to understand for the why part. Mainly here you could just remember is that they can attack any and every organism, right? There are examples of virus attacking virus also. So yeah, they can attack themselves also. They are a very complicated organism which is under immense study, has been and is always be under immense study because of the unique characteristics they have, right? So they attack different organisms and based on the organism they alter their machinery or they alter their functions, right? They take up that cell's machinery, they alter their genetic information, their functions, and adjust accordingly to that organism. So because of that, the virus can mutate very fast, or mutate is basically it can develop variants, right? It can develop new varieties where some part of it is different, right? So yeah, because it does not have membranes, all this is possible, and because of that, as you can see right now, as this is a classic example, it is wreaking havoc, right? Because of this reason. So yeah, this is what this entire paragraph means, that basically the importance of membrane can be illustrated with the example of viruses. So imagine that if you actually have a membrane, you can stop everything, or you are contained, or you cannot cause any damage or anything to other organisms, right? Okay. Okay, this is a small flowchart for you to just, so this will help when you're revising the chapter or when you're just going through this PPT as a recap, right? So this has all the important organelles listed here. So you could just look at this and check whether you've gone through all of them and or not, right? Simple for that. First thing is our endoplasmic reticulum. So basically, it is a large network of membrane-bound tubes and sheets, right? This is how it looks like. There are two types, rough and smooth. So it looks like long tubules, round or oblong bags, which are called vesicles. There are two types, rough and smooth. Yeah. Oh, this cartoon isn't working. Why is that? Okay. The rough endoplasmic reticulum has ribosomes attached to its surface. Right here, this is a ribosome which is attached to the surface for an example, right? And that's why it is known as rough. It gives a rough kind of feel to it, isn't it? And the main function of ribosomes is they are protein manufacturing sites, and the function of endoplasmic reticulum is to transport this protein to various parts of the cell. As you can see here, they are long tubules, which means they're basically something sort of a passage, right? And they extend right from the nucleus to like one end of the cell. So there are these really long passages which are used for transporting stuff, mainly proteins, right? Okay. And then you have smooth endoplasmic reticulum. Again, this is also an example to show you where it is located. So immediately next to the nucleus is always rough, and after the rough is the smooth endoplasmic reticulum, right? The smooth endoplasmic reticulum has different functions. It helps in the manufacture of fat or lipids, right? Rough is involved in proteins transport, versus smooth is involved in the manufacture and transport of fat and lipids, right? Some of these proteins and lipids help in building the cell membranes, right? And that process is called membrane biogenesis. So anything where you are creating something is known as genesis, right? Biogenesis is basically it's a biological component. That's why it's called a biogenesis. And what is it creating is a membrane, hence the term membrane biogenesis, right? Okay, and oops, sorry. Yeah, some other functions also are there, like yeah, they could function as enzymes or hormones. And in the liver cell of vertebrates, the smooth endoplasmic reticulum helps in detoxifying many poisons and drugs. Right? Okay. And these are some of our functions. Like I said, it's a huge pathway, right? Because it extends from one end to the other end of the cell. So it's like a pathway or a channel for transporting materials, mainly proteins, right? From various regions of the cytoplasm. Or it also acts as a cytoplasmic framework for providing a surface for most of the biochemical activities of the cell. This point basically means is that you suppose this is your endoplasmic reticulum, right? So for enzymes or activities to occur, they need a surface, right? They need a spot where they can attach or where they can stay and perform, finish their activities, and then go ahead or move on, things like that. So basically the endoplasmic reticulum is that site where these substances can take a break, or they can just lodge themselves, and or find a catalyst here to facilitate the reaction, right? Depends on what exactly that substance does, what is its function, it depends on that. So cytoplasmic framework basically means it's like a structure. A framework is basically something you call a plan, or a you know a jotted down flow chart, that okay, this will have these components. Then, like for example, a flow chart of the cell, like a cell will have nucleus, cytoplasm, and plasma membrane. In the cytoplasm there are organelles, right? That's the framework of a cell. Similarly, this ER is basically a framework for the biochemical activities of the cell. So either substances can come to the ER and finish their work, or they are while they are transported through the ER, they are able to finish most of their work. Either of the cases, right? Okay. Again, we are not really having that much time, so I will skip this video. You can watch it after class. Difference between rough and smooth endoplasmic reticulum. Again, it's basically whatever is being mentioned in the previous two slides itself. Uh, are there any extra points you could just refer to your book, or you could add if you can't remember these ones? You could refer to other points and write those also. Okay. Next component or next organelle is the Golgi apparatus. Frequently asked question is the scientist who named uh, the Golgi apparatus. So his name is Camillo Golgi, right? It comes as an MCQ usually, right? They give you examples. They'll mix up all these scientists' names that Robert Hooke, Leeuwenhoek, Golgi, and maybe Schleiden or Schwann or some other random scientist and ask you to identify which of them discovered or described Golgi apparatus or Golgi body, right? The other name is Golgi body. So this is what it consists of. It's a system of membrane-bound vesicles. Vesicles are basically flattened sacs, or what you can see here, right? Or suppose you have a balloon, right? If it's a balloon not in the inflated state, so it's just like you brought a fresh packet of balloons and you're going to blow air in them. You know, the balloon looks something like this, right? Deflated balloon. That's a flattened sac. That could you could remember it like that. Okay. Arranged approximately parallel to each other as shown in the diagram, right? In stacks called cisterns. So the stacks are called cisterns, right? And each flattened sac is called a vesicle, right? Okay. Remaining things are extra information like the cis phase and trans phase is extra information. You don't really need to remember those things at this stage. But this was a very clear diagram, so I picked this up, right? Okay, these membranes often have connections with the membranes of the ER and therefore constitute another portion of a complex cellular membrane system. Basically, they're trying to say is that sometimes these Golgi bodies are found connected to the ER. So with the ER, they become another long pathway, right? So imagine that you are building a train, right? So this becomes another coach. It becomes another long passageway, something like that. Okay. So what are the functions? Like I said, it's a pathway. Any pathway will usually involve transport, right? And the additional function here is packaging. So you can see in this cartoon here it's showing. So the proteins, once they enter, they are packed and they are transported out, right? Storage, modification, and packaging of products in vesicles. One important function is the formation of lysosomes, right? Lysosomes are formed here. In some cases, complex sugars may be made from simple sugars in the Golgi apparatus, right? This is a point where which might help you later on, not right now, because here you need to know what are simple sugars, what are complex sugars, right? So sucrose, fructose are generally known as simple sugars, whereas glucose, etc., are complex sugars. So basically, it will convert or it will combine various simple sugar molecules to form the complex sugar molecule. Basically, that building stuff here. Okay. Here, if you remember, the first 2 points is sufficient: storage, modification, packaging, and formation of lysosomes. Right. Okay. Yeah. And then next we come to what are lysosomes. Again, a very frequently seen question. That why are lysosomes known as the suicide bags of the cell, or what are the functions of lysosomes, right? Either of the two. One question is always there on lysosome. Okay. What are lysosome is rarely asked. Usually, why is it known as the uh, suicide bag or the waste disposal system or the janitor of the cell? That is asked, and what does it do is asked, right? Okay. So membrane bound sacs filled with digestive enzymes, right? Keep the digestive enzymes part in mind. As you can see here, the red one is the lysosome, right? And it comes and it attacks this bad thing and it just eats it up, right? Yes, like I said, functions waste disposal system helps to keep the cell clean by digesting any foreign material as well as worn out organelles, right? And how are they able to do this? Because they contain very powerful digestive enzymes, right? So sometimes when the cell has gone beyond repair or it has been damaged to a large extent, so it engulfs the entire cell itself. It can eat up the cell itself, right? Yeah, like is mentioned here in the third point, that during disturbance in cellular metabolism, that is basically when the cell, when there is too much wrong inside the cell, like the cell has gone beyond repair, or too many foreign organisms have entered, or some parts of the cell have broken down, right? The lysosome may burst and digest the entire cell. That's why it is known as the suicide bags of a cell, right? Okay. Next, you come to mitochondria, the very famous one. It's always asked as uh, uh, the powerhouse of the cell, right? It's known as the powerhouse of the cell. So, main question you might get here is, why does it have folds? Okay, or what is known as the powerhouse of the cell? That is the only question which comes. Function also rarely asked. Structure rarely asked. Diagram sometimes, yes, because not that complicated to draw, so sometimes yes, but mainly asked as what is the powerhouse of the cell, right? Why is it called a powerhouses? Because ATP generation takes place here. ATP is known as the energy currency of the cell, right? Because it's involved in producing energy, it is known as the powerhouse, right? So it is a two membrane structure, right? Outer porous and inner deeply folded membrane. These folds increase surface area for ATP generating reactions. Basically, because of this fold, it's able to produce more ATP. That's all it means. Right, the functions, like I mentioned in short, it's given in detail here, right? ATP, you must know the full form, again asked as an MCQ, or they might just ask you as a one marker question, what is the full form of ATP? Yeah, one important fact to note here is that mitochondria have their own DNA and ribosomes. Another organelle which has its own DNA and ribosomes is plastids, right? Uh, yeah, that's why they are called strange organelles. Maybe if you take up bio later on, you will hear that mitochondria and chloroplast were actually independent cells before. They were these unicellular kind of organisms like bacteria or so, right? And they actually evolved into current stage mitochondria and chloroplast. So because of that, they have their own DNA and ribosomes. They used to function as independent entities before. They are direct descendants of bacteria, right? Okay. Again, this is something when you go ahead or when you take up bio further, you will read about this in detail, right? Okay. Yeah. So plastid is the other one which has its own ribosomes and DNA, right? Okay. And plastids are only found in plant cells, right? There are two types, chromoplasts and leucoplasts. Chromoplasts are basically colored. Chroma stands for color, okay? Plasts are short form for plastids, right? Chromoplasts are colored, so usually the most commonly found chromoplast is chloroplast, which has green color chlorophyll pigment, right? Again, all are a bit difficult terms if you're reading it the first time. So chlorophyll is the pigment, chloroplast is the type of plastid, which is a type of a chromoplast. Okay, yeah. And the other type are leucoplasts, which are white or colorless, and they are mainly involved in the storage of starch, oils, and proteins. Again, this is also asked as an MCQ question in the HOTS MCQ, like a higher order level MCQ, right? Yeah. So this is the top diagram is a basic structure for chloroplast. Sometimes they ask you to draw the diagram. Again, it's a relatively easy diagram, so that's why they ask you to draw it. Where you have thylakoid. So each individual sac is called a thylakoid. It's arranged in stacks called grana or granum. Single stack is called granum. Multiple stacks are called grana. These grana are connected by these network called lamellae, right? Okay, and the matrix or the equivalent of chloroplast in a plastid is, the equivalent of cytoplasm in a plastid is stroma of the matrix, the fluid in which this is suspended, right? And this cartoon is just showing you that chloroplast, it takes the light from the sun and it makes glucose. Basically, it's performing photosynthesis, right? Yeah, and sometimes they also ask you the difference between chloroplast and mitochondria because they are very similar organisms, but they perform different functions, right? So chloroplast makes glucose, whereas mitochondria breaks down that glucose to give you ATP, right? Or a process known as respiration, right? So these are the differences which are mentioned here. Okay. And next you have vacuoles. Vacuoles are storage sacs basically, right? They are small in animal cells and large in plant cells, right? Where plant cells it can go up to 50 to 90% of the space, like it's shown in the diagram here, right? And what do they contain is basically they're full of cell sap. Cell sap is nothing but a liquid. Okay, a sap is like kind of a sticky liquid. So this liquid is found in cells, so it's called cell sap, right? Again, what are the functions? They mainly are used for storage and to maintain that rigidity. So here again there is the term turgidity, right? So if you go back to what we learned, how is a cell staying like this, right, without collapsing? So this is suppose your cell membrane. The way it is staying like this without collapsing is half, or you could say partly attributed to vacuoles, right? This is your vacuole. I've shaded it in red, right? It helps in maintaining that structure of the cell, helps it not collapse. Okay, and it mainly stores substances. The substances could be water, as you can see in this comic here, water, nutrients, amino acids, sugars, various organic acids, or proteins, right? Anything depends on what type of a cell it is, it would store different substances. And in amoeba, the food vacuole contains food items the amoeba just consumed, right? So the amoeba basically engulfs food. This is how it looks like. When it sees a food particle, let's say this is your food particle, let me make it yellow, right? So when it sees that, it will extend its arms like this and it would take up the food particle, right? So, and yeah, and then it becomes like this. So the food is inside, this is the nucleus, and the food is inside the amoeba. So this then becomes a food vacuole, is basically what they're trying to say, right? In some unicellular organism, it expels waste and excess water from cell. Okay, let's skip this because we have a little bit part left, right? Yeah. We'll quickly see this video. I want to show this video because cell division is a little complicated topic at this stage, right? So cell division is basically the process in by which new cells are made, right? And there are two types, basically mitosis and meiosis. They are different based on the number of daughter cells and based on the number of gametes, or the number of, you could say, ploidy of or chromosome number in the daughter cells, right? So mitosis usually occurs in somatic cells or body cells, and the number of chromosomes in the daughter cell is the same. Like if you see this cartoon here, if the parent cell has 46 chromosomes, it divides and the daughter cells also have 46, right? Versus in meiosis, the parent cell starts at 46, it undergoes division twice, right? Again, maybe in next year or later on, you would break down meiosis into different types of division, where one of it is actually mitosis, right? And in the end, so it undergoes division twice and gives rise to four daughter cells, and each of them have half of the number of chromosomes of the parent cell, right? So 23 is like 46 by 2. right? That's how you remember it. Chromosome, right? One more thing I wanted to show before we see the video. So basically this is the difference, right? Mitosis occurs in body cells, right? Or skin cells, stomach cells, etc. These are examples of body cells or somatic cells as they are called. Versus meiosis occurs in gamete cells. So it will only occur in these two. There are no other examples, right? They make gametes, right? Or here mitosis make body cells, right? This is like a Venn diagram where the this part here is showing what is common between them. Is basically they are both involved in making new cells. That's all that is common between the two. Okay, let us come back to our video. Let's see the video. Okay.
Have you ever thought of cloning yourself or regenerating some of your organs? But being humans, we can't just.
Are you able to hear the audio? Is it fine?
Ma'am, it's breaking a little for me.
Okay, it is not clear for you. What about the others? Breaking. Acha, breaking. ठीक है. It's breaking. Okay, okay, then let me not get into it. I will finish. Yeah, I'll just take you through these one or two diagrams. We have finished with the thing. Maybe you can watch the video uh, after class, right? Okay. I've already shared the PPT on the group, but in case you've not got it or you missed it, I'll just share it right now also so that you could go through it right away, right?
Yes, ma'am, can you share because I've lost it.
Yeah, yeah, I'll just share it again. Not an issue. Okay. Yeah, and these are some of the diagrams you need to know, like plant cell diagram and animal cell diagram is usually asked. Sometimes it's a 5-marked question where you are supposed to draw and label at least 5 to 7 structures, not just organelle. You could label nucleus, cytoplasm, etc. also, right? So I'm saying structures. Right, so this is one of the diagram which is easy. I think it's the same one from NCRT. It's the easiest one to draw. If you go to see, the others get a little complicated. So yeah, and you could pick out any few, just pick out few important ones which you can remember and label them. So practice this diagram, and this is an animal cell diagram. Again, this is not for drawing, this is for understanding the organelles or the structure of animal cell diagram. For drawing, you could use the one from the chapter notes or the one from NCERT, right? Those are good enough. Or it's the same like plant cell, you just need to make it in a circular shape, don't make a rectangle, and don't draw a cell wall, right? And no chloroplasts. 2-3 things you need to remember, right? Okay. Yeah, I like this. This is there in your chapter notes. This is a very important difference or important diagram which tells you which structures are there only in animal cells. So these are only in animal cells, these are only in plant cells, and these are there in both, right? So only animal cell may lysosomes, many small vacuoles, nucleus is usually in the center, and this is round, right? Why do plant cells lack lysosomes is again an important question which is asked as a give reason, right? Maybe we'll have a give reason or a practice session on all these chapters next week where we'll just go through questions, right? uh, yeah, we'll just go through questions. So okay, that time we'll take up those questions where uh, why is lysosome not there in plant cell, etc. is there. So both is a wide list. If you remember which are there only in animal and only in plant, I think both you will excluding those are there in both, right? So whichever way you feel easy to remember, you can adopt that, right? Plant cell main two are chloroplast and cell walls, right? Those are the main two only present in plant cells, right? Okay. Yeah, these are all concept maps, or you could say flow charts. These will be helpful when you are remembering or when you are revising the chapter. So this is just to check if you've covered everything. So you can just read this and be like, okay, do I know what is osmosis? Do I know the definitions? Do I know the types of osmosis? Okay, what is diffusion, etc. Right? Okay. Exocytosis, endocytosis. Again, this is just a small bit. I think is there in the NCRT pertaining to amoeba, right? So how endocytosis basically means taking in stuff inside the cell, and exocytosis is basically removing stuff outside the cell, right? I just drew the amoeba diagram, right, where it took up food. So the same thing happens in reverse when it wants to remove waste products. That's exocytosis. Okay, ठीक है. Again, this is again a concept map for you to revise or check you've completed all three or not, or you remember all the organelles or not, etc. These are all helpful during revision times. If you are going to read it now, it might not make any sense, right? Okay. And these are additional video resources. So I've also mentioned what video is for what. So like anytime when you are bored reading theory, or when it's become too much theory, just open one video and watch it. It'll be fun also, and you will revise something also, right? Okay. ठीक है. Yeah. So that is all about. Yeah, one more picture. Oh yeah, there is one more picture of just a cartoon, which, yeah, is just saying what the different organelles say if they could talk, right? So nucleus is the brain or the controller. That's why it says everyone do what I say, right? Versus endoplasmic reticulum does most of the work, even Golgi. That's why they are saying that. Lysosome destroys everything, right? Vacuole is just storage, so it feels full, right? That's a pun here. That's on fulfilled and full. Yeah. Mitochondria, haha, no, because it has its own ribosomes and DNA and proteins, right? So that's why it's rebelling against this. That's all. Okay. This is just like a cartoon, just for fun. Okay. I wanted to ask one thing quickly. Have I given homework before from this chapter or no? I haven't. I can't remember.
Yes. No, ma'am.
Yes? No, I haven't given.
I don't remember. Ma'am,
I think
Suja says,
initially you had given one, but recently I don't think so.
yes, ma'am, you have given. You have asked us to do this third living cell. And yes, ma'am.
did I ask you to do theory-based question and give reasons? Have I? Okay. Okay, let me check once. I must have written it somewhere. I also can't remember, so I thought I'll ask you all. If I haven't given, I will give it to you now, so you can do it over the weekend, right? Just simple. I won't give too many questions. Just the theory-based and give reason questions. The first questions or practice one questions from cells, so that when we have practice session next week sometime, we can start level one, right, directly, or we can take up doubts and then start level one. You know. So let me just check. If I haven't given, I'll give it to you. If I have, I will skip it. Okay. ठीक है. Okay. Anybody has any doubts or anything you would like to discuss? No. Okay.
No, ma'am.
No. Okay. No. Okay. ठीक है then. If no doubts, then cool. I will share the PPT with you on the group right now. So maybe you could watch the videos, right? They're all tiny videos. I haven't really included any long videos. They are just 4 to 5 minute long. So yeah, only one video I think which is about yeah, this overview of the chapter. The overview of the chapter is 30 minutes long. I think so. But they have explained everything, A to Z, whatever you need to know from this chapter or whatever you need to remember is given here. So in case sometime you're revising or you don't want to read anymore, just watch that video. It's good. It's really good. Okay. ठीक है. And in case when you are watching videos, if you come across any interesting video which you feel others should also watch, you can always share on the group, right? Anytime. Okay. ठीक है. Okay, then let's wrap up. ठीक है. See you all. See you all next week during practice.
Thank you, ma'am.
Okay.
Bye, ma'am.
Thank you, ma'am. Bye, ma'am.
Thank you,
Bye.
ma'am.
Thank you, ma'am. Bye, ma'am.
Bye bye.